Hi everyone, I'm Amelia, a PhD student from the University of Granada. In this talk, I will present the results of a project in collaboration with the Delft Bioinformatics Lab. The title of the talk is Unsupervised Protein Embeddings Outperform Handcrafted Sequence and Structure Features at Predicting Molecular Function. The automatic function prediction task using machine learning algorithms has shifted from using handcrafted features to automatic learning these features. The features can be learned by a complex model which requires a large amount of training samples to be properly trained. Because of the close relationship between protein sequence, structure, and function, we hypothesized that by combining sequential and structural information, we can perform better at the task of predicting the protein function. We focus on the molecular function ontology, the most correlated to sequence and structure, but we also made some small experiments in the biological process and cellular component ontologies. The prediction models can be trained in a supervised way, guiding the training by some predefined values or classes. This process is dependent on the prediction or classification task and requires enough labeled proteins, which are limited to a few thousands in this case. On the other hand, unsupervised training is guided by the data itself and does not depend on a prediction or classification task. In this case, we can use the millions of unlabeled proteins that are stored in the UniProt to learn a complex feature representation. Then, these pre-trained features can be used for the specific task, either by model fine-tuning or transferring these features to new models. The language modeling of protein sequences is an emerging and promising field. Several deep unsupervised models have been recently made available, which use different neural network architectures from the natural language processing field to learn deep representation of the amino acid sequences. In this project, we consider a publicly available pre-trained model from the ROST lab to struct embedding representations for protein sequences. Their model, called CQBEC, is based on the ELMO model from the NLP field, which is trained to predict the next amino acid in a sequence given all the previous amino acids. This model was trained using 33 million of proteins from UniRef50. As there is a presentation from the ROST lab in this track, we are only providing some details about their model architecture here. It is composed of 3 connected parts, an embedding layer to embed each amino acid in the sequence, and 2 layers of bidirectional LSTMs to learn the context. As stated in the paper, the 3 outputs can be summed up to obtain an embedding vector of size 1024 for each amino acid. In addition, we can get a fixed size protein level representation by averaging all amino acid embeddings over the sequence dimension. In order to represent the protein, we used both sequence and structure information. We compare the ELMO embeddings, both amino acid and protein level, to 2 handcrafted sequence representation. One corresponds to the 1 hot encodings of amino acids, which protein level representation contains the frequency of each amino acid in the sequence. The other contains k-mer counts, whose dimensionality has been reduced to match the ELMO embedding size. We also structured several features from the 3D structure, including the secondary structure and backbone angles, which we compare to the pre-trained deep fold and contact map representations. The protein structure is defined by the position of the amino acid atoms in the 3D space, from which we can extract the secondary structure state of each amino acid and the angles formed by consecutive amino acids. To obtain a rotation invariant representation of the protein structure, we compute the Euclidean distance between each 2 pairs of beta carbon atoms. The resulting distance map is a symmetric real-valued matrix of variable size L times L, where L is the protein sequence length. From this map, we extracted the default features using a pre-trained model of protein structures. By applying a distance threshold, we obtain the contact map representation, which is a binary matrix of size L times L as well. To process it, we can use either a 2D CNN or a graph convolutional network, which can process sequence and contact map information together, as shown in the recently published preprint by the Richards Cornell lab. Here, we include this animation to better understand the differences in processing the contact map. The contact map can be viewed as a binary image, in which we can apply 2D convolutions to extract spatial patterns from the map. It can also be interpreted as the adjacency matrix of a graph, where each amino acid is a node and edges represent amino acids that are in contact, each row or column of the contact map. We used the first approximation of the graph convolution from Kipps and Welling, in which A corresponds to the contact map and X contains the amino acid level features. In the experimental setup, we compared different protein representations and several prediction methods. The KNN, logistic regression, and multi-layer perceptron classifiers work on the protein level features, average ELMO embeddings and one-hot encodings, k-mers, or deep-fold. The 1D CNN and GCN models work on the amino acid level features, either ELMO embeddings, one-hot, or structural features. The GCN model processes the contact map as a graph. Also, it can process only contact map information by using the node degree of each amino acid as feature. The 2D CNN model also works on the contact map, but viewing it as an image. We intended to use simple model in all cases, so the neural network architectures contain few layers, one for the MLP, two for the 1D and 2D CNNs, and one for the GCN. The input of the models that apply convolutions is of variable size in the protein sequence dimension, either amino acid level features or contact maps. So a global pooling operation is needed after convolutions to get a fixed size representation before the fully connected layer that provides the posteriors for each go down. To train and evaluate the models, we created 3 datasets with protein structures from the PDB, one for each ontology, that are annotated with non-computational codes. We divided each dataset into train, validation, and test sets. We had around 8K proteins to train the model and 1K proteins to decide the optimal parameters for the models. For evaluation, we used a test set of sequences with a maximum of 30% sequence identity with the train and validation sets. In this dataset, we evaluated around 250 terms for molecular function and cellular component ontologies, and 1K for the biological process ontology. We first showed the molecular function results on the 30% identity set test subset. We calculated the term-centric ROC-UC for each classifier using different protein features, either protein level, amino acid level, or contact map. The error bars show the 95% confidence intervals that were estimated using 1000 bootstraps. We also compared with the BLAST and naive baseline. As you can see, the ELMO embeddings in orange outperform the rest of protein representations, and they are closely followed by the pre-trained default features that are in green. The best ROC-UC result is given by the MLP method using the protein level ELMO embeddings. We can also notice that in the case of the GCN classifier, the contact map information provides a performance boost when using a simpler representation as one-hot encodings in blue. In addition, the contact map alone, which is in gray, can predict molecular function better than the BLAST classifier, but worse than the pre-trained ELMO and default representations. We further analyzed the contribution of the contact map information in the GCN models when the ELMO embeddings are present. To do so, we changed the adjacency matrix from the contact map to a random matrix with the same number of edges. We also used the identity matrix, in which the amino acid features are processed without combining with the neighbors one in the graph. As can be seen, the performance when using ELMO embeddings as node features is the same for the three adjacency matrices, but it gets worse for the one-hot encodings when changing the matrix. This suggests that the main performance of these GCN models comes from the use of a powerful node representation and not from the contact map. Therefore, if we would expect similar results if we were to replace the true contact map with a predicted one when using the ELMO embedding representation. The superiority of the ELMO embeddings compared with other representations is also seen when evaluating the biological process on the top and cellular component on the bottom ontologies. We then analyzed the performance of the models at different levels of terms specificity. To do so, we used a larger dataset with sequences from SwissProt, evaluating 441 terms from the molecular function ontology. We compared the ELMO embeddings to one-hot encodings in sequence-based model only, both protein and amino acid level. As in the PDB dataset, the method that provided the highest ROC-AUC was the MLP with ELMO embeddings. By comparing these models, we saw that a simple KNN model with ELMO had a larger mean ROC-AUC than the 1D-CNN when using one-hot encodings. We plot the differences in ROC-AUC between both models as a function of term specificity given by the path length to the root in the GO graph. The correlation between the two measures is on the top of the figure. For each level being, the number of terms is provided on top and the median ROC-AUC difference on the bottom, which also corresponds to the horizontal lines. We found that the 1D-CNN models perform better for more specific terms. The rest of the models that use ELMO embeddings had a better performance across all levels in the GO graph. This showed that the unsupervised ELMO embeddings contains a general function description, but supervised training is needed for the more specific terms. We also made a small experiment on the SwissProt dataset by combining the layers of the ELMO model in different ways. As a reminder, the ELMO model is composed of three parts, one embedding layer and two bidirectional LSTMs. If we average each output over all amino acids, we get a fixed size representation from each part. We compared three ways of using the information of each layer. In the first one, we summed up the three layers, as we did in previous experiments. In the second one, we only used the 1024-dimensional hidden state of the last LSTM layer. And in the third one, we concatenated the embedding layer and the hidden states of the two LSTMs in a vector of 3072. The logistic regression results show that there is a performance drop when using only the last LSTM output. It does perform better when concatenating the three outputs in a higher dimensionality vector. The differences are statistically significant, but the effect sizes are very small, as you can see from the scale on the y-axis. Finally, we perform experiments on the CAFA3 dataset, using ELMO embeddings as feature representation and the KNN logistic regression, MLP, and 1D-CNN classifiers. We compare with the CNN of the DeepGo Plus method, which uses one-hot encodings, and with the top-ranked methods in the CAFA3 challenge. As you can see, our MLP method would have ranked 5th out of 146 methods. Also, a simple KNN using ELMO ranked 44 for biological process and 10 for cellular component. The preliminary CAFA4 results may have been released by the time of this presentation, so we will have a clearer view of how this model ranks against the current state of the art. In summary, in this project, we show the superiority of pre-trained features over handcrafted protein representations at the task of protein function prediction. We saw that the language model ELMO is useful to predict the protein function, not only for molecular function, but also for the other two ontologies. This complex representation cannot perform using simple classification models. Also, we did not see a performance boost when adding protein structure information to the ELMO embeddings, which is helpful when combined to simple representations as one-hot encodings. The ELMO embeddings contain information about general functions, but further supervised training is helpful for more specific terms. Finally, as language modeling of proteins is a promising field, future work could provide a systematic comparison with other protein sequence embedders in the function prediction tasks and also other protein-related tasks. This concludes my presentation. I would be happy to answer any questions in the chat, but first I want to thank my collaborators, my advisors, the funding sources, and of course, all of you for your attention. Thank you so much.